It is my pleasure to introduce Dr. Deepa Gautam, an accomplished scientist whose work spans some of the most intricate aspects of platelet biology and cancer research. Dr. Gautam completed both her master's and PhD in biochemistry at Banaras Hindu University, where she explored the impact of neuromodulatory molecules on platelet signaling. Following her doctoral work, she moved to Thomas Jefferson University in Philadelphia as a postdoctoral fellow, delving deeper into the molecular mechanisms regulating the activation of the key platelet integrin alpha-2b beta-3 and its interaction with CIB1. Currently, Dr. Gautam is a postdoctoral researcher in the Department of Hematology at Brigham and Women's Hospital, Harvard Medical School. Her ongoing research focuses on understanding the roles of distinct platelet subpopulations in cancer, a field that holds tremendous promise for advancing precision medicine. Please join me in welcoming Dr. Deepa Gautam.
Can you see my screen?
Yes, ma'am.
Okay, perfect.
Yes, Deepa, I think you have to just make to presentation mode. Yeah.
Thank you. So today I'm going to talk about platelet subpopulations and their potential role in cancer progression. We know about platelet, but now, like from decades, people have started talking about platelet subpopulations. So I cannot introduce myself better than whatever introduction the organizing team has made. So I will just skip the slide and introduce you guys with the platelets and to keep everyone on the same page. So platelets are colorless cell fragments in the blood and it is produced in the bone marrow. So this was discovered first in the blood by an Italian scientist. I'm sorry, it's very hard to pronounce their name. So it was developed in it was discovered in 1882, and its size is ranges from 2 to 4 micrometer. Platelets have the lifespan of 7 to 10 days in circulation, and the normal platelet count is 150K to 450K per microliter of blood. And the main function of the platelet is like hemostasis. It maintains the hemostasis of the body. And when we see the platelet structure, it doesn't have its nucleus or DNA. So when the megakaryocyte produce these platelets, it gives all protein and RNA to these platelets, and it comes in the circulation. And as it has to perform its function, so it has lot of mitochondria and it has different granules which stores different factors that help platelet to perform their functions. So basically what happened when there is any damage in the blood vessel, platelets comes into action and it forms the platelet plug. So to stop the bleeding, which is known as the hemostasis. So platelets are known to play important role in hemostasis, but from several years now it has been shown that it not only play role in hemostasis, but also in other functions like inflammation or neurodegenerative disorders. But we in our lab we are more focused on cancer and metastasis. So we are trying to understand the role of platelets in cancer. Platelets, they are known to play important role at different stages of the cancer. For example, it interacts with the primary solid tumor and helps its growth. Once the tumor cells, it comes into the circulation, now it has to escape the immune cells. So tumor cells are very smart. It make a shield of platelets around it and to just hide from the immune cells. And not only this, but also platelet help the tumor cell to locate to the other uh, part of the body, and which we call as metastasis. So platelets are known to play important role in all these three steps. What happens when platelets they are produced by the megakaryocytes? They come in the circulation, and newly formed platelets, which are young platelets, have lot of RNA and protein. But once it like spend some time in the circulation, it loses its RNA and protein, and then they are known as old platelets, just like human, young and old. And once they get old, it is cleared from the body. So based on this, platelets has been classified into young platelets and old platelets. And these young and old platelets, they differ on the basis of the protein that is expressed on the receptors. And the most important difference between these two subpopulation is the RNA content, which we exploit to physically isolate these young and old platelets from the blood. And it has also been reported that these different kind of the platelets that is present in the circulation plays important role in different kind of diseases. So our lab is focused on the breast cancer, like we work on the breast cancer. So we were just curious to know whether these platelets are like young versus mature platelets, whether their quantity differs in the breast cancer mouse model as compared to the wild type. So PYMT is the breast cancer mouse model, which develops breast cancer with age. And in our lab, like this was not done by me, but some of my previous lab members who showed that these mouse model have more young platelets in circulation as compared to the wild type. So the question was why they are more. If they are more, there will be some reason that they are increased. So when I joined the lab, my question was like whether these two different type of platelets in the circulation, they are different, whether they respond differently to the agonist. So I performed a very simple experiment where I compared unstimulated platelets and the activated, that is the platelets when have different receptors. When they get the stimulation, they get activated. So I found that these, when I'm telling like TO high, thiazole orange is it is a chemical that binds to the nucleic acid. And as I mentioned that young platelets, they have more RNA, so they will bind more to the thiazole orange. So when I'm saying TO high platelet, it means they are young platelets. And as we can see in the heat map, like these TO high young platelets were more activated as compared to the mature platelets. So I also like looked into the literature and found that platelets are also classified on the basis of functions. Like there are aggregatory platelets, procoagulant platelets, coated platelets, and secretory platelets. So these different type of platelets that is in the circulation, they differ on the expression of the receptors, and they also bind to the different proteins that is present in the blood. Out of all these, this procoagulant platelet is the most activated platelets. Like if it is present, it means the body literally is not in the right state. So my question was, when I started my this research, my question was like the current blood report gives you the platelet count and the mean platelet volume, but it never tells you about what kind of platelet is circulating in the body. Because in the disease condition, when the platelets are activated, they get cleared. And megakaryocyte, which produces platelet, get the signal like, okay, platelet count is down, so make more platelets. And platelet count is replenished, but what kind of platelet is getting replenished, we never know. And many patients who are on antiplatelet agent, like you might have heard around you, like some people are on aspirin, or like cancer patient or the stroke patient, but they never respond to these antiplatelet agent. So from here, my main research idea came like, why are we only measuring platelet count or mean platelet volume? but we also have to measure, like see which kind of platelets are circulating in the body, and then we can target those specific subpopulation. So the first step in my research was to design a panel or the tool, like how I will study different subpopulation. How, if I will get a blood sample, how will I know, like how will I quantify different subpopulation? So for that, I made one antibody panel which stains different receptors on the platelet, and I designed this antibody panel keeping in mind that I can cover all important surface receptors that is involved in different functions, like in inflammation, in cancer, or some receptors that is important in neurodegenerative disorders. And after this antibody panel, one of my co-author, he helped me in high dimensional analysis. So he used all the Python libraries or to helping me in analyzing these different subpopulations. And when we combine this antibody panel and this high dimensional analysis, we were able to see 4 different kind of platelet subpopulations in the circulation. So resting platelets are the physiological platelets, which are totally normal platelets. When the platelets are little bit activated, they are called prime platelets. And when they are very activated, they turn into the aggregatory and the procoagulant platelets. So these pro-aggregatory and the procoagulant platelets, these are like really activated one. Like it will have all activation markers upregulated. So once we made this panel and this software, I was um, I was like, I should use this and see whether we can see some interesting thing in the patient. And the first group that we chose was the mesothelioma cancer patient. So mesothelioma cancer patient, mesothelioma is the cancer of the lining of the internal organs. The most common is the pleural mesothelioma, that is the cancer of lung lining. So these patients, they are known to be facing lot of thrombosis, like heart attack after surgery. And when you will see the blood report of these patients, you will see like every blood report is normal, like they will have normal coagulation factors and the platelet count. But why are they clotting more? So we asked a question whether platelets or its subpopulation, they are playing important role. And also these patients are on antiplatelet agent, but they never get benefit from these antiplatelet agent. So first, to confirm whether these patients are really clotting, we in the lab we checked the clotting parameters. So briefly, I will introduce it. This is the thromboelastography, which measures the formation of the clot and the strength of the clot. The formation of the clot depends on the coagulation factors, but the strength of the clot depends on the platelets. So we were more interested in the strength of the clot, whether they are upregulated or not. So we found like the red trace is the mesothelioma. So this is known as the amplitude. If this is higher, it means all the platelet dependent factors are upregulated. So in the mesothelioma, we found like these mesothelioma had stronger clot as compared to control. And these are different parameters that depend on platelets. So from these graphs, it was confirmed like these patients have like platelets which are super activated. So now the question was like, I was like, okay, we should use our panel and see whether we can see different subpopulation in these mesothelioma blood. And then I used my panel and the platelet profiler. And we found that these patients, the mesothelioma patient, had more procoagulant platelet in circulation as compared to the control. So, I also compared like TO low is the mature platelet and TO high is the young platelet. So TO high and TO low platelets in the mesothelioma, they were more activated. So this says like our panel could catch different subpopulations, and now it's time like um, we can target, we should not, I mean, my idea behind this is like we should give subpopulation specific medication to improve the efficacy of the drug. Because and now we have started using this panel and this analysis in different patients, like the patients who are facing like multiple myeloma patients, then also the GI cancer patients, and we are seeing like these patients have different kind of subpopulations in circulation. And most of the patients that I'm talking about, they are on antiplatelet agent, but they are not getting any benefits. So I will conclude in a way, like we are trying to develop a platform where I can study different subpopulations and then give a subpopulation target, like specific target medication. And this is my team with whom I work. So my PI, like Batinelli, she supports me a lot with whatever idea I come to her. She's like, okay, let's do it. So she's very supportive. And I work with my collaborator, this Dr. Bueno. He is the mesothelioma surgeon here who provides me with all the meso samples. And Becky helps me with like I am detecting different subpopulations, but how these subpopulations are connected or correlating with the severity of the disease. So she's on the clinical part and she's trying to connect the severity of the disease. So this was my small presentation. And let me know if you have any question.